Using your muscles to move or lift something. What exactly causes that to happen? Our purpose for this video is to explain the process of a muscle contraction. Before going into the process, let's first take a look at the structures involved in the muscle. Skeletal muscle tissue is connected to your bones by tendons, and this is what actually causes a muscle flexion or contraction to happen. This whole structure here is called a fascicle, which simply means a bundle. In this case, it's a bundle of muscle fibers. These circular units here are the actual muscle fibers called the myofibers. They are responsible for your muscle movements and contractions. Surrounding every myofiber is the sarcolemma. It is like a thin protective sheath that encloses the myofibers. Within the myofibers are several cylindrical structures called the myofibrils. Inside the myofibrils are hundreds of protein filaments called thick and thin filaments. These filaments are arranged in patterns on the sarcomere, cross-section illustration of a microfibril, also known as a muscle cell. This covering here represents the sarcolemma, which surrounds the muscle cell, and it serves as a protective sheath and forms the membrane connections between the sarcomeres through passage of the T-tubules. This area here is the sarcoplasmic reticulum, which is a specialized endoplasmic reticulum in the muscle cell. Within each of the muscle cells, you will find multiple nuclei and the mitochondria. Now we will take a look at the different zones located on a sarcomere. The sarcomere is the structure in muscle fiber responsible for your muscle contractions. Here we have the A-band. The A-band extends along the total length of the thick filaments. This area here is the I-band. The I-band is an area between the A-bands. In this area, there are only the thin filaments. Each I-band extends across two sarcomeres. This area here is the H-zone, and it is located at the center of the A-band on each of the sarcomeres. In this zone, there are only thick filaments. The H-zone, the A-band, and the I-band all provide information about the state of a muscle contraction based on their locations on the sarcomere. These here are what we call Z-discs. The Z-discs are regions on the sarcomere that separate the neighboring sarcomeres.
Muscle contractions are the results of action potentials. When the impulse is given off, the action potential travels down the axon. This action potential causes calcium channels located on the axon to open up. The calcium ions begin to pour into the axon. Inside, there are synaptic vesicles that are filled with acetylcholine, or ACH, a neurotransmitter. The calcium moves the synaptic vesicles down the axon until they reach the end of the bulb, where using exocytosis, they fuse with the membrane and push the ACH into the synaptic cleft. Once out, the ACH binds to special ACH receptors on the motor end plate. Once two molecules of ACH bind to one receptor, the motor end plate opens up sodium channels, allowing sodium ions to flow into the myofiber, making it more positively charged, causing a second action potential.
This is the changes in ion flow through voltage-gated channels during the depolarizing and repolarizing phases of an action potential. At the resting state, the cell membrane is more positive on the outside than it is on the inside. Sodium ions are floating outside of the membrane, and potassium ions are floating inside of the membrane. Once an action potential occurs, the cell membrane becomes more negative on the outside and more positive on the inside. This causes the sodium channel to open and sodium ions to flow inside of the cell membrane, causing the depolarizing phase.
Once the action potential has caused the sarcoplasmic reticulum to release calcium into the sarcoplasm, the calcium then binds to the troponin on the thin filament that allows troponin to move off the actin, which then exposes the active site for the myosin head to bind on the actin.
This is a muscle in a relaxed state. Once the tropomyosin has been removed from the myosin binding sites, this is when the muscle contraction begins. The myosin heads have ATP binding sites and the enzyme that hydrolyzes ATP into the ADP and a phosphate group. Once the cross bridges have been formed, a power stroke begins, thus opening the site where the ADP is still bound to the myosin head. The myosin head rotates and the ADP is released. This is the action that initiates a force. The thin filaments start working their way towards the center of the sarcomere. As the myosin heads continue to attach themselves to the binding sites, they pull the thin filaments in towards the center of the sarcomere. Once they reach the center of the sarcomere, this is when you receive a full muscle contraction.
This is a summary of the events of contraction and relaxation in the skeletal muscle fiber.
First, the nerve impulse arises at the axon terminal of the motor neuron and triggers release of acetylcholine. ACH diffuses across the synaptic cleft, binds to its receptors in the motor end plate, and triggers a muscle action potential. Acetylcholinease in the synaptic cleft destroys ACH, so another muscle action potential does not arise unless more ACH is released from the motor neuron. A muscle action potential travels along transverse tubules, opens the calcium release channels in the sarcoplasmic reticulum membrane, which allows calcium ions to flood into the sarcoplasm. Calcium binds to tropopin on the thin filament, exposing the binding sites for myosin. The contraction power strokes use ATP myosin head, bind to actin, swivel, and then release thin filaments are pulled towards the center of the sarcomere. Calcium release channels in the sarcoplasmic reticulum close, and calcium active transport pumps use ATP to restore low level of calcium in the sarcoplasm. Calcium release channels in the sarcoplasmic reticulum close, and calcium active transport pumps use ATP to restore the low level of calcium in the sarcoplasm. Tropopin-tropomyosin complex slides back into position where it blocks the myosin binding sites on the actin. Then the muscle relaxes. As you can see, muscle contraction is a pretty extraordinary process that involves many structures and molecules, and it is necessary for the movement and the functions of the human body.